What is up, y'all? Kevin Kuhn here from Athlete Factors. This is the Athlete Factors podcast. My guest today is Stephen Machek. How you doing today?
Good. How you doing?
Very well. Thank you. So, the quick synopsis of why you're on today is to talk about some new research within the realm of powerlifting. So, I guess take it away. Tell us a little bit about yourself. You're a PhD student, so tell us a little bit about that, and your personal, your athletic background, your academic background, and all that.
Sure, I'd love to. Like you said, I'm a third year PhD student at Baylor University. I'm working under Dr. Darren Willoughby, I initially got into exercise science because I was actually an overweight kid. So I was an overweight kid, and I was kind of tired of kind of the stigma behind that. So I wanted to make a change. So I started losing weight the way that everyone wants you to lose weight, which is to run a whole bunch and starve yourself, which didn't turn out very well. But in the process of it, I learned all the things that I was doing wrong, and I learned about nutrition. And so I decided to do my bachelor's degree at San Francisco State University in dietetics. Fast forward like 4 years, something like that. If you know anything about dietetics, you apply for an internship after you've completed the 4 years. And I didn't get in the first time. And one of my main weaknesses was that I didn't have much clinical experiences. The clinical stuff has never been my forte. So I had a couple of different options. One is to get more clinical experience. The other is to go ahead and do a master's of something that would help complement kind of your interests. And I was the founding member of the sports nutrition subcommittee in the General Dietetics Association. So I'd always had a really interesting passion that I had developed for resistance training, and specifically powerlifting. That was the lens that I found was the most interesting. After doing resistance training for about a year, I just realized that I really enjoyed the powerlifting movements. And so I decided to do a master's in exercise physiology, and I worked under Dr. Jimmy Bagley, also at San Francisco State University. After doing that for a while, I realized that I really enjoyed the research aspect of it a whole lot. And I can talk about my thesis too at some point. Is actually kind of similar in the sense that some of the research we're going to be talking about, where we looked at an elite powerlifter, a middle-aged powerlifter who had been doing anabolic steroids for several decades. And we looked at his myonuclear domain. I can talk about that kind of stuff, and his fiber type. It was really, really interesting work. And I also realized that the research aspect also gave me an outlet or acted kind of like a filter to hang out with some people that had some really similar interests. So after graduating there, I decided that I wanted to do my PhD, and I wanted to do something that could kind of synergistically employ the nutrition and the exercise components, because I don't know if you've been to too many universities and seen those individual departments, but usually they don't have curriculum that intersect very well. They generally tend to stay very separate. At least that was my experience. So I wanted something that kind of encompassed the whole experience. So that's why I reached out, and I eventually found Dr. Willoughby, who does a lot of research, obviously, in resistance training, but also has a huge emphasis on the dietary supplements or nutritional interventions that I think are really neat. I really think that finding these minute mechanisms behind these different things is such a cool way to go about researching some of these supplements. Obviously the industry takes advantage of that, but some of these things actually have some space to work, and they can end up being really novel and really cool. So I think that's, I guess the last part I'd like to talk about is the powerlifting. So I have been powerlifting. I'm not great at it. I'm very mediocre at it, but I think that's why I've pursued a lot of trying to learn about it and trying to research about it. It's just because it gives me another outlet to kind of convey that passion. So I coach some powerlifting in my spare time. I basically try to get everyone that I meet that becomes my friend into powerlifting, got my wife into powerlifting. Nice. My best man at my wedding was I met through powerlifting, and it's just been a big passion of mine that I've been trying to feed forward to people whenever I meet them. So yeah,
that's awesome. Well, it's a fun sport to watch.
Debatably.
Well, I've never competed, but I've seen some, some meets or some competitions, and um, it can be very, very entertaining. I
Yeah,
mean,
not
at
bench.
least
People usually leave during bench to go to lunch.
I've seen. Granted, this was, and we kind of mentioned this earlier, the Arnold, before we started recording, but I remember seeing uh, a guy just gushing blood from his nose,
Yeah,
hitting a world record squat.
yeah,
So it's stuff like that'll pique your interest,
yeah. Deadlift and
perhaps.
squat really get people going. You start to see the faces turn purple and oh man, people puking. You ever see that? People puking as they're
Actually, yeah,
deadlift.
yeah. Oh
I've
yeah,
never seen it in person, but
yeah.
the urination aspect is really common. I remember the first time I was watching a powerlifting meet, and then it was a straight stream of urine forward. And the great thing is that the person that was lifting that weight, like they didn't care. They were getting that lift, that third attempt deadlift, and she was just as stoked after getting that. And
Yeah.
that was really neat. I don't know how she would have acted if she hadn't have gotten it, but
I mean, we act like that doesn't happen in other sports.
oh
It
yeah.
does.
I was just listening to, I can't remember who was talking. It was another it was an article or something like that about the rates of urinary incontinence and in certain women's in certain sports, and it was those numbers were mortifying. So
It happens.
yeah,
You just, sometimes that
no one talks about
intra-abdominal pressure just gets to
it.
a
Yeah,
point where
yeah, and no one talks about it either. So
yeah.
that was
Well,
fine.
I will not be conducting research in that area in the future.
No, yeah, I think that's beyond my scope. I'm gonna go with that one.
Hmm. Yeah. Awesome. So, uh, let's get into this article that was just recently published. So tell us a little bit about it. The title actually I found extremely interesting, but just because it took me back to neuromuscular exercise physiology class with
Mhm.
Dr. Willoughby. We were learning all about myosin heavy chain isoforms and um, different muscle fiber types and things like that. So talk us through that a little bit, and we'll kind of jump into this research article.
Sure. So the general premise was kind of like as a powerlifter myself, I don't have a huge focus around. I'm interested isn't necessarily biomechanically related. I've always had a kind of a general affinity towards more of the cellular stuff, and so I know a lot of papers have been published that talk about a lot of the predictors for powerlifting performance. So whether that be certain leverages, so like brachial index, how long your arms are, or how long your torso is. There's a lot of papers that kind of look at that stuff, but nothing that's really trying to examine at the molecular level what are some of the predictors that we can find. And like I said, I had done my master's thesis on fiber type, and that was part of it. And so what I wanted to see was what are the main molecular predictors. So I was thinking about some of the main characteristics or main adaptations that resistance training and specifically powerlifting can impart.
Mm-hmm.
And so I wanted to see which one of these things can actually predict powerlifting performance specifically related to the Wilks coefficient, which is, for anyone that's not familiar with it, it's a normalized score based on the total amount that people are lifting. So their best squat, their best bench press, and their best deadlift, and then they normalize that to the person's body weight. So obviously, if you have an entire meet of people, the person that is the biggest, because there is that linear relationship with how large someone is to a certain degree, how much they can lift, that then it would just end up with the biggest person essentially being able to lift the most, or at least the larger people being able to lift more than the smaller people. So this is a really good way to kind of normalize that, so that how well can this person lift relative to how big they are as a person. And I think it's just as cool to be able to see someone who's 70 kilos lifting, like a 1,600 pound total than like a 120 kilo guy lifting 2,000 pounds plus.
Mm-hmm.
So that was the general impetus behind the reason why we did that particular study. I don't know if that answered your question in particular.
Yeah, yeah, for sure.
Yeah.
So let's jump into I guess some of the specifics. for people who aren't really familiar with kind of any of that sort of thing, theoretically, can you talk a little bit about the different fiber types and what, at least in theory, would be more beneficial for powerlifting?
Sure. Yeah, so I think most people will know that there are slow twitch fibers and there are fast twitch fibers, type 1s and type 2s respectively. And when we try to relate those to performance or some of the characteristics inherent to them, we know that type 1s are slow twitch, so they typically will be more conducive towards kind of low intensity, long duration types of activities. So your quintessential endurance runner, marathon runner, that type of individual, because those fibers are characterized by more mitochondria, larger mitochondria, more capillary density for more gas exchange to be able to get more oxygen into the mitochondria.
That
More
boring oxidative stuff.
Yeah, exactly. That's the stuff that I don't like. Exactly. And then there's the fast twitch stuff. Now that's the fun stuff. That's where we tend to start to associate higher intensity, shorter duration types of activities. So powerlifting, Olympic weightlifting, sprinting. Yeah, those are your kind of quintessential, very high intensity, short duration types of things. And those are characterized by having, a more intricate network when it comes to the T-tubule system for generating action potentials to go down, and a larger sarcoplasmic reticulum, so better capacity for calcium handling in that regard. And then typically we have a larger amount of the contractile elements in type 2 fibers. So we're talking about actin and myosin for a lot of that specific tension development, because that's where we know a lot of that actual cross bridging is happening with the sliding filament theory. Mm-hmm. So I mean that's encompassing type 1s and type 2s. But I think if you go a step further, you know that there's type 2As and there's type 2Xs. And we have this spectrum. It's a spectrum that's ever evolving. We have the type 1s on the slow side. We have type 2Xs that are on the very opposite end, very fast. And then we have type 2As that exist kind of more so in the middle, that represent fibers that are fast twitch but also oxidative at the same time. Mm-hmm. And with different developments of techniques, and I don't know if I'm going to get a little ahead of myself here, there's different ways to measure fiber type. So we have histochemistry, where we can actually stain for myosin ATPase, which is the enzyme that's actually on the myosin head that can actually cleave and utilize cycle through that ATP. So we have that method. We can stain for that, and we can basically subjectively look at these fibers and see, well, this one's darker, that one must be a type 1, that one's pretty much white, that one must be a type 2X, mm-hmm. or a type 2A. And
can I pause you for a second?
Yeah, go for it.
This part is not relevant to the conversation at all. However, I think it's extremely, like, it's gonna be stuck in my head. Have you ever heard anyone call it AT pace? Because that bugs me.
Yeah,
it's ATPase. Can we all just agree that it's ATPase?
Yeah, I mean, I hope we could.
It's ATPase. Thank you for saying that.
Yeah, I'm glad I didn't mess up. So if I say something that you're like, is he saying that right? Just let me know. That way I can correct myself after the fact.
Awesome. Everything's sounded really good so far. So
okay.
I didn't mean to jump in there and cut you off.
Good. You're good.
So, yeah, I hope I didn't stop you mid thought. I can't. It just jumped into my head, and I was like, I have to get this out. So,
you're good.
Let's see. So, in theory, it's going to be better for powerlifters to be more towards that type 2A or maybe even type 2X end of the spectrum.
So that's a really good portion that I'm trying to get to as well, is the idea that, especially because I teach undergraduate ex-phys to a certain degree, so I teach the lab and I also teach a little bit of the lecture. I'm working my way in to try to teach the lecture because they don't really let us do that all that much. Mm-hmm. But I always ask people because I want to see where their opinions are. So if you look at the spectrum, you would assume that someone like an Olympic weightlifter, someone like a sprinter, you would want the fastest possible fiber type, right? So you'd want the phenotype that can contract the fastest. But what we're actually seeing is that with 2X fibers, Number one, 2X fibers are extremely rare, like less than typically an athletic population the general population will have less than 5%, less than 2% of pure type 2X fibers.
Wow.
The problem is with 2X fibers for athletic populations is that they are so fatigable. So they fatigue so fast that they're not really relevant for actual activity, right? So they can generate a whole lot of force and they can generate it really fast. They are still the fastest force generating. They have the most power, right? So force times velocity. They have the best ability to generate that force fast, but they fatigue so fast that they can't maintain that force over time. So the 2As are actually where we start to see a lot of the power athletes starting to shift their fiber types towards, if we look at this spectrum. So basically, there are two real athletic fiber types. There's type 1s and there's type 2As. And basically, depending on the intensity of the exercise, it's going to deviate one way or the other. I don't know if you want me to talk about hybrid fiber types or if you know anything about hybrid fiber types.
Go for it, man.
Okay. So there's the methods that I was talking about. There's immunohistochemistry where we can look at the myosin ATPase, and then we have homogeneous SDS page. I mean, as a side note, you can also look at other things like different enzymes using those staining techniques. But for the most part, for the sake of immunohistochemistry, it's ATPase. And then we have homogeneous SDS page or homogeneous sodium decyl sulfate polyacrylamide gel electrophoresis. I try to say that every single time that I say the word SDS page because otherwise I'm going to forget it, but it's a tongue twister. Where basically what you do is you take the muscle, you grind it up, you put it into this machine that basically uses an electronegative current to push the proteins, and you can separate the proteins out into bands and then quantify the proteins based on the molecular weight. So you can look at the percentage in a given sample, how much of this is type 1s, how much of this is type 2s, and how much of this is type 2As or type 2Xs. So the good thing about that is that we can get a more quantitative look at it, a more objective look at the fiber types, or at least the composition of this entire thing, how much of the space is being taken up by 2As, type 1s, 2Xs. The good thing about the staining was that we could individually look at all these different fibers, and we could actually differentiate between the three different fiber types, but also we could see that there were some fibers that didn't necessarily align with the paradigm, so that it was like we have white, we have black, but what about different shades of gray, right? Like what do we actually classify those as? And so my master's advisor, Dr. Bagley, and a lot of Dr. Galpin's work has been looking at the single fiber level. So what they do is you'll take this big biopsy, and then you'll individually pick out these fibers, kind of like you would pick out noodles of spaghetti that are kind of bundled up together. And what you can do is you can take those individual fibers, put them in, and do the same gel electrophoresis technique, that same SDS page technique. And what they can see is we have the classical spectrum or continuum of type 1s, type 2As, and type 2Xs, but we also have isoforms that exist in between. So we have type 1/2As, we have type 2A, 2Xs that exist here. We even have type 1, 2A, 2Xs, so they have all three. Wow. Or one of the guys that I was doing my master's with, he was working with end-stage renal disease patients and trying to do resistance training with them. And he was one of the only people that ever saw type 1, 2Xs. So it's less of a spectrum. It's kind of more like a circle, honestly, where we have shifts that are occurring, and sometimes like kind of arbitrary shifts where it doesn't seem like it's along the continuum, but we have some that actually represent like disease states. So the only time that we ever really see 2X fibers is with people like spinal cord injury. Those are where we actually see like the pure type 2Xs. I mean, there was one instance where there was an elite sprinter who had like 25% or something like that, but that's one person. And then the hybrids, I think the one main thing I wanted to get across about the hybrids is the hybrids exist because they are kind of there because they want to differentiate. So they're not really good at anything, but once you start training, they will transition one direction or the other. So if I have 20% type 1, 2As, the hybrid in between, and I start doing aerobic training, they're going to start shifting towards that type 1. If I start doing resistance training, they're going to start shifting towards the 2As. And we have that on a spectrum. So we can assume sometimes if we have someone who has a percentage of type 1s, a percentage of 2As, if that person's 2As, they might shift if he starts to do or she starts to do more aerobic types of activity. And they'll go through that spectrum. They'll go from 2As to 1, 2As, and then to 1s eventually if the training stimulus is robust enough or long enough, if that makes sense.
Yeah, that whole idea, like learning that that was a possibility where you can shift some of your muscle fiber type one way or the other depending on the specific training stimulus was extremely interesting to me when we started learning about this when I was at grad school, and it was kind of around this time where like some of the questions that you're asking in this study, like
Right.
we've been thinking about for a while, but like at the time we all assumed like, oh well, if you want to be extremely explosive, if you want to exert the most amount of force, then you want to shift as far as you can towards type 2x.
Right.
So it's super interesting to me that like that's maybe actually not super accurate, just because and like now thinking about it, like it makes sense, like because in order for that shift in any sort of fiber type to occur, it has to be based off a training stimulus. And in athletics or in any sort of sporting type training, like it always comes back to a specific level of conditioning and repetitive bouts of training, right? So that's going to shift things towards a little bit more towards that type 2A position, right, or the aerobic side, the type 1. So I think, man, that's really interesting stuff.
Yeah, I guess one thing I should note is that those hybrids are pretty much the first thing to start to go. So some people genetically have more hybrids than others, and that's where they're going to start to differentiate. The next level is going to be those 2As to 2Xs. They're innervated by the same motor neuron, so they're both alpha 1 motor neurons. So some of those phenotypical changes that can occur are a little bit, at least in my mind, they're a little bit easier because we're still working with the same motor neuron. But when it comes to type 1s to type 2As, it's possible. It's just harder for that change to happen over time. It needs a more robust stimulus. And with humans in general, I mean, we can look at animals all day. We can look at animals from birth till death, but we know that animals don't even express the same fiber types as they express different fiber types. So in humans, it's not ethical to take a baby, and look at their fiber type over the years. That's not ethical. But some of those changes we don't know for sure is where I'm trying to go with that, especially when it comes to some of the higher intensity stuff. We have a lot of literature that's looking at the aerobic end of things, but when it comes to how different athletes respond from the power side of things, we're very heavily under-researched in that regard.
Gotcha. Another interesting thing that I think is, I guess, at least somewhat relevant is like the function or the purpose of specific muscles. Some are a little bit more, let's say, locomotive.
Yeah.
Some are a little more postural.
Right.
So, or I think some people call them tonic and phasic. Right. So, postural muscles, you probably want a little more towards that aerobic side because they've got to work all the time. Whereas, these prime movers, let's say the glutes, quads, hamstrings, calves, you want those muscles to be able to exert a lot more force.
Definitely.
So I think, there's can you talk a little bit about the different, let's say, percentages of the different fiber types within different muscle groups?
Yeah, I mean, I think some of the classic stuff, like you already talked about, with postural muscles primarily being type 1 fibers because of the nature of how often we're using them. They need to be oxidative. They also they need to turn over a little bit more because we're using them more constantly.
Mhm.
Things like the gastroc being more fast twitch, things like the soleus being more slow twitch. And the reason that we actually use the vastus lateralis more often than not is not only because it's a good indicator of leg function, so we can look at that, we can determine, what's the relationship with activities of daily living and ambulatory activity, things like that, but also because it's generally about a 50-50-ish. It generally represents about like a 40-60, something like that. So it gives us a good idea of where fiber types are going because we know that particular muscle can respond really well, to training. And
Mm.
that's one of the reasons why we use the VL. I mean,
For
over time, I think
biopsies?
for biopsies, yeah, I should have
Gotcha.
qualified that. Yeah. I mean, there are other muscles that we've used for biopsies before, not me and our lab personally, but I've heard of calf biopsies, which sounds miserable. Yeah, sounds miserable. I've even heard of trap biopsies, if you've ever
Wow.
seen that before.
I have not.
Yeah, so there's some research, some lines of research that have actually looked at. So the traps actually have a higher androgen receptor content. So if you look at like anabolic steroid research, I think it was Coddy in 1988. I think it was something like it was Coddy or Erickson, one of those main researchers that were looking at powerlifters who were taking anabolic steroids. And so they did a trap biopsy and looked at the androgen receptor content in there and the fiber types there. And I can't remember what the fiber type was specifically, but one thing that makes sense to me is that the traps are, because of that androgen receptor content, they're probably going to be more susceptible to hypertrophy. If
Mhm.
we were going to have a particular fiber type in the traps, we would want the one that probably is a little bit more plastic to growth. So we would probably have a higher 2A content there.
Mhm.
Yeah. So I don't know if that answered your question, but
No, that's very interesting. That's
yeah,
cool stuff.
you usually
Um,
get sidetracked with things that are cool.
this is so awesome, man. This literally takes me back to grad school. I love it. Like,
Yeah.
it's so
Well, I
fun.
mean, I'm there, so like I'm just you're living vicariously through me. I've been in grad school for, the last 5 years pretty much. So
That's awesome. So you said you're this is again a little bit off topic, but you said you're a third year.
yeah, I'm third year, so I
So
just
how much
finished
is
my.
left?
So I was told that I could finish it in May, but like we're expecting, my wife's pregnant and she's due in
Mm,
February. So
awesome.
I have that, and then I'm trying to get all of that done by May and apply for jobs. And money is not very so like funding opportunities are a little bit more sparse nowadays because of the pandemic,
Hmm.
and all those things just kind of led me to want to push it off for another year. So
Mm.
the objective for me is to get my dissertation done pretty early next semester, and then I can just spend pretty much an entire year just milking out the writing, waiting for money to
Best way
come
to
in,
do it.
applying to jobs, and being able to actually sit down and not write it up in a night, because I
Yeah.
mean that's pretty much what I would. If I've already missed the point to apply by now because most of them are over in October.
Mm.
So it's actually kind of like a weight off my shoulders because I was trying to deal with writing up a proposal for my dissertation. Also we're doing pilot work right now for the dissertation.
Hmm.
I can talk about that later too if that's something that you're interested in.
For sure.
Um, yeah, yeah. So trying to get all that done and trying to apply for jobs, it just sounds like so much. It's horrible.
That's so crazy, man. Well, it's good that you have some time that you don't have to rush it. That's kind of nice.
Yeah, yeah. I feel kind of bad because I know that my wife wants to get out of here, and I want to move on too, honestly.
Hmm.
Um, but it's good to be able to. I'm comfortable. Like, I've gotten the motions down. I know what a day-to-day kind of thing looks like over here.
Yeah,
So just having a little bit of time to kick back, even if I'm kicking back with a baby screaming in the background. I mean,
that's
you
fine.
know,
There's
yeah,
nothing wrong with that.
yeah.
That's awesome.
Yeah.
So, we talked a little bit about kind of muscle fiber typing, and we've touched a little bit on bioenergetics. But can you give us a little bit of a primer with regards to the different bioenergetic systems, just because I think that's going to play an important role in discussing a little bit more of your research.
Sure. Yeah. So from the perspective of some students who have probably taken some of these kind of rudimentary exercise physiology classes, we have our phosphagen system, or our creatine phosphate adenosine triphosphate system, ATP-CP system. Immediate pathway is another way that we talk about that, which is basically just the system that we use to recycle ATP really quick from creatine phosphate. So when we break down ATP, we have inorganic phosphate and ADP. So that creatine phosphate can then, for lack of a better term, lend that phosphate over to the ADP and give us more ATP to work with. So we have that. We have the glycolytic system. So both of those, the immediate and the glycolytic system, we would kind of consider to be our anaerobic system, so we don't need oxygen to utilize them. Overall, the reactions are much faster as well. So with glycolysis, we can take advantage of glycogen that's already stored within the muscle cell per se, and then we can create ATP, a very small amount relative to the aerobic systems that I'll start talking about. But we can derive ATP from that, and then we can also derive lactate. So we can convert pyruvate into lactate, which we can't use as a fuel necessarily right there within the fast twitch fibers that are probably utilizing this system. But what we can do is we can shuttle that out with hydrogen ions that are the breakdown product of ATP hydrolysis, and then we can shuttle those to oxidative tissues like the heart, the slow twitch muscle, the brain, where we can actually reconvert that back to pyruvate. And then I guess that segues really nicely into the aerobic systems that typically use oxygen, or do use oxygen. We are able to take glucose and fat in many cases with a lot of the longer duration, lower intensity type of stuff, and we can yield a heck of a lot more ATP from that. The downside is that it just takes a little bit of time for that oxygen to perfuse. It takes a little bit of time for the oxygen to go from the ambient air to your lungs to the blood to the given tissue that is demanding that oxygen. So the anaerobic systems are especially important for kind of the beginning of exercise, where we're trying to kick on exercise, but they're also really beneficial for the higher intensity types of exercise, where that ATP demand is, I guess, overcoming or exceeding our ability to meet ATP production through oxidative means.
Gotcha. So I've always found it really interesting, and maybe at least initially I thought it was very convenient. Perhaps now it's not so. Perhaps it's an oversimplification, right? So we've got 3 muscle fiber types, and we've got 3 main bioenergetic systems, right? So at least my initial understanding was, oh, this is simple. The ATP PC system lines up with Type IIx,
Yeah.
and then
I wish it were that convenient.
Wouldn't that be nice? An
That would be
anaerobic
fantastic.
glycolysis, Type 2A, and then the oxidative, boom, Type
Yeah.
1.
Yeah.
Done. Not so simple, but there is, however, some overlap with, I guess, those systems, right? So,
Yeah, definitely.
so that being said, when it comes to, we got to bring it back to powerlifting.
Yeah.
What's the bioenergetic system that we're trying to leverage or take advantage of the most when it comes to powerlifting?
Right. So I guess I should start off by saying that all the energy systems are always active. It's just an emphasis on one more than the other, given this particular type of activity or how long that activity is, the intensity of that activity. So typically powerlifting, if we're talking about the competitive aspect of powerlifting, for the most part, they're going to be in the 1RM percentage range of anywhere between like low or high 80s, low 90s, all the way up to 102, 106% of someone's previous 1RM. Sometimes, if someone's extremely new, you can kind of surpass that range. But for the most part, the length of an individual maximal attempt isn't going to surpass the capacity of the ATP-CP system or the phosphagen system. So it's going to be probably less than 10 seconds, 15 seconds, and that's usually where people say is the limitations of the phosphagen system is it lasts anywhere between 5 and 15 seconds. You might be able to eke out a little bit more if you naturally have a little bit more, or if you take like creatine supplementation to augment your stores. So I guess when I was talking or trying to rationalize this for the study, it wasn't so much that we were trying to say that, well, powerlifters are going to be exceeding the need on that system while they're in their meets per se. But it's interesting to see if the total amounts of creatine phosphate that are inherent to the individual might be a better predictor. So like genetically, do they have more creatine phosphate, and does that potentially augment the training that they've done to that point to get them so that they have a higher Wilks coefficient, so the relationship there? Or is creatine phosphate or the creatine levels that you have, does that relate towards the overall training capacity? Because not every training set that you have is going to be under 15 seconds. A lot of these are going to be 3-5 rep sets, sometimes upwards of 6, maybe 10, depending on what you're doing in a particular training block. You're not only going to be taking advantage of the immediate pathway, you're going to be taking advantage of glycolysis. And then what are you taking advantage of during your rest periods? You're taking advantage of, you're taking advantage of aerobic systems. Yeah, exactly. But the idea too is that with a lot of powerlifting training, you're taking things submaximally. So you're in a certain percentage range, but you're not going to failure in a lot of these sets, especially not with the main movements that you're doing, the competition movements. I know that if you told me to take 75% of deadlifts and take it to failure, I would tell you I'm probably not going to do that because I'm going to mess something up in my back, especially if I'm experienced enough to remember what 75% feels like. That I wouldn't be able or wouldn't want to take that for as many reps as possible or to failure just for safety reasons. So that kind of references me back to one of the studies that I referenced in my introduction, where they looked at equivalent load. So they looked at the leg press. So it's not a 1-to-1 here, but they looked at leg press, and they saw that there was a higher depletion of creatine phosphate when they had submaximal, or they didn't go to failure, rather than when they took all of those sets to failure. So same load, one didn't go to failure, one had a set number of reps, the other went to failure. The one that didn't go to failure utilized more creatine phosphate. So I'm thinking powerlifters are probably putting a little bit more stress on this system. It'd be interesting to know if there are some adaptations that come with that as well.
Gotcha. That's really interesting.
Yeah. Hopefully that all made sense. Might have talked in a circle.
No, I think I'm following. So that kind of sets us up quite nicely to then dive headfirst into, like, we kind of have the framework, we've got the foundation. So talk us through your study. What were the variables you were looking at? What were the methods?
Right.
Let's dig into it.
Yeah, definitely. So we wanted to obviously look at powerlifters, and we wanted to see what made them different, or what made them different with respect to how could we compare some of these things to their performance as per Wilks coefficient, being our kind of surrogate performance marker, or surrogate skill marker. So in order to do that properly, we couldn't just compare within group. We wanted to get a good, accurate control. So we found sedentary controls, kind of just students around campus or whoever we could find. And then the powerlifters that we recruited were from the local Texas area. And we set an arbitrary cut point at 300 for our Wilks coefficient. And there really isn't any solid definition of what makes a powerlifter good or not when it comes to the Wilks coefficient. But overall, if you were to look up kind of meet results, you would see that overall, like generally people that are experienced tend to be at least above 300. So we set that as an arbitrary cutoff point to make sure that we weren't getting just extremely inexperienced powerlifters. And because we were using that database, they needed to have competed at least once. So that was kind of our threshold there. They needed to be actively training in powerlifting, so at least 3 times a week doing the powerlifting movements.
Mm-hmm.
They needed to have competed within the last 12 months, and they needed to have at least a 300 Wilks coefficient. In retrospect, I mean, you could have set that higher, but because there isn't really anything that would dictate what a good or a bad powerlifter is, it's, saying 400 might have limited my pool quite a bit more. So I just kind of wanted to see if some of these adaptations had already been incurred by the point that they had that experience enough to be higher than 300 and have competed already. Mm-hmm. So what we looked at is it was a cross-sectional study, which means that we didn't tell them to really do anything. We didn't have an intervention per se. So we didn't control their training. We didn't control their diet, although we did have them record their diet. But basically they would come in after having not trained for 48 hours, just because we know that some of the stuff that we were looking at might be affected in some way or another. And we took blood from them and we took a biopsy. And it was a particular method of biopsy. So it's the fine needle aspiration method, which uses a much smaller, kind of minimally invasive, as minimally invasive as a biopsy can be, from their vastus lateralis. And we compared some of these markers, and I'll go over the markers in a second, to those sedentary controls.
Is that the one that kind of shoots
Yeah.
out?
Yeah, did you ever get to see one of those?
Oh yeah, I got to help out with a lot of them.
Oh, fantastic. It's great. It's fantastic. So,
super cool. I was the distractor.
Oh, nice. Okay.
That was my
holding
job.
on to the foot and asking them asking them what they're about to eat later, things like
nice. Trying to tell them jokes and all that stuff. Mm-hmm. That was fun. But I was usually not paying attention to them. Like I'm talking to, I'm not looking at their face. I'm like looking down. I'm like, this is awesome. That leg's cut open or going in.
Yep.
I loved it, man.
Yep. But it's not as invasive as a Bergstrom needle.
Big needle.
You take a scalpel, you go in there. I mean, it's in terms of the discomfort, it's not magnitudes higher than the fine needle, but in terms of the soreness that you get afterwards, at least in my own experience, because I've had both done on me. I tried to do a glute ham raise on the glute ham raise machine one time after getting a biopsy done with the Bergstrom needle. That's not happening. That's not fun. I can do squats after a fine needle aspiration, but I can't do glute ham raises after getting a Bergstrom done. It's horrible.
No, I can imagine. I mean, you're like cutting out some steak, man.
Yeah. I mean, with the fine needle, I mean, you can the pros to the Bergstrom that you get a whole bunch of tissue all at once.
Mm-hmm.
The cons are that, you get that big open gaping hole. But with the fine needle method, you just have to go in so many times that it makes the process go a lot longer than some people are comfortable with. And especially if they're in their head about it, With the Bergstrom needle, it's in and it's out. It's like if you committed to it by the time that lidocaine's in your leg and that needle's in your leg, I mean...
One and done.
Yeah, exactly. One and done. Yeah.
That's hilarious.
Yeah. So some of the markers we looked at, so we looked at fiber type, and it's worth mentioning that we actually didn't do the fiber typing in-house. So with the muscle we used, we actually sent the tissue off to Dr. Andy Galpin's lab. It was a recommendation from my advisor, Dr. Bagley, back at SF State, and Dr. Willoughby was fine with the whole collaboration aspect of it, which is really cool to me because it's like I got to marry two parts of my grad school experience. So we sent the tissue off to them, and they did the entire Homogen SDS page where they just basically break the tissue up. And the reason we did the Homogen and not the single fiber that I mentioned way back then is because the tissue is so small, it's really difficult to individually tweeze out fibers from it. So that is a particular, I guess you could say a limitation, but I can talk about why it's not a limitation a little bit later on. So we did that for the fiber typing, which gives us type 1, type 2A, and type 2X composition. So it doesn't tell us the percentage of fibers per se, but it does tell us, so in this bundle of fibers that we mash up, how much of this is type 1, how much of this is 2A, and how much of this is 2X. We also looked at muscle total creatine and serum total creatine. And then we also looked at the transporter, the SLC6A8, which is responsible for transporting creatine that is either consumed exogenously or produced endogenously into the cell. So one thing I should mention about that is typically what you'll see is that type 1s tend to have more of that transporter than type 2As, or type 2Xs for that regard. And then the last marker that we looked at was creatinine. So if anyone's ever gotten their blood tested, typically you'll see a creatinine marker, and that's usually used as kind of a surrogate marker for renal function. But in this sense, what we're looking at is in totality of it, it's a better marker with respect to the breakdown of creatine. So it's a metabolite of creatine. And that's just giving us a really good global perspective of what's happening with creatine metabolism. We have blood, we have the transporter, we have it in the muscle, and then we have the breakdown product.
Nice.
Yeah. So I think that's all the markers that we took. Obviously, we run statistics on that, but then to look at how, in particular, some of these things are affecting Wilks coefficient. We ran correlations between myosin heavy chain 2A content because we know that relationship with performance and specifically like strength athlete performance. And then we looked at the total muscle creatine content and related that to Wilks coefficient. So those two things in particular. I guess one thing that I totally forgot to mention was that pretty much no female powerlifters have ever been researched ever. So our group was divided. So we had 12 powerlifters. 6 of them were males, 6 of them were females. And these are actually the first 6 female powerlifters that have ever been researched before.
No way. That's awesome, man.
Yeah. And it was really cool because I don't know if you have subscribed to like this idea, but typically when we think about fiber types, we think that women tend to have a higher oxidative fiber content, so more type 1. So I wanted to see can the training kind of overcome that like it did in one of Dr. Andy Galpin's previous research studies in weightlifters, where they actually showed that the most experienced female weightlifters at the world-class level actually had more type 2A fibers than the national level males, which I thought was really cool. Yeah.
That's super interesting.
Yeah. So we're looking at all those relationships. It made it very statistically irritating, but
but that's because you're the first one doing it.
Yeah. Well, let me know next time not to make it that statistically complicated.
Oh man, yeah, you're blazing the trail.
Yeah, I guess that's fair.
Yeah.
Gotta show some love to the ladies, I guess. So.
For sure. No, that's cool stuff. So, what were some of the results? What were some of the conclusions?
Yeah. So with the fiber type stuff, we saw that the powerlifters had higher type 1s and 2As relative to the controls, and we were seeing that relatively higher content was kind of at the expense of 2Xs. So actually only participant, only one male in the study had any 2X fibers whatsoever. And he actually had a relatively high percentage of them. It was like 20%. But pretty much no other powerlifters had any type 2Xs, or no powerlifters. I should qualify that a little bit better. As opposed to the control side, I think we had an average of somewhere between 20 and 30 of the 2Xs.
No way. That's so crazy to me. That blows my mind.
Right, which is exactly, so it's counter to what a lot of people would believe, that general spectrum that people believe if you're powerful, you want 2Xs, right? So that's not at all the case. These are sedentary controls, I should qualify. So they were sedentary based on the ACSM guidelines. So they were exercising less than 30 minutes a day, 3 days a week, over the last 3 months. So they were very sedentary, and they had the highest percentage of type 2Xs.
Wow.
Yeah. One really interesting finding was that our females overall, so regardless of group, actually had higher 2A content. So already we're kind of challenging some of those beliefs, but the really interesting part of that interpretation was so it's not just the powerlifting females, but it's our females in general. The Stronger by Science group actually did an analysis on this paper in particular, and they made a good point. They just mentioned how we had one male in the powerlifters who kind of threw off some of that 2A content because of that 2X that 2X percentage he had, And then in general, a lot of our males had a higher 2X percentage and a lower 2A percentage. So at a group level, it kind of made sense that that the females would have a higher 2A percentage. I don't think there's anything particularly mechanistic about that. If the female powerlifters had a higher content and not the controls, we might be able to infer that there was some type of skill level difference between the two. So maybe we didn't control for skill or experience well enough to be able to detect that or control for that. But that wasn't the case. So we kind of think that that's kind of a more aberrant finding in that regard.
Gotcha.
Yeah.
Why do you think and this just kind of popped into my head, why do you think it is that it was thought that women would tend to have a higher type 1 fiber content, and now that we're getting some data showing that, it's actually type 2A, like, do you have some sort of either evolutionary perspective of why that is, or like do you have some sort of theory behind the rationale there?
It's a good question, and I would be kind of pulling at straws to try to come up with something that's coherent. But I mean, evolutionarily speaking, I would assume that men were more of the hunters. I think historically, so they would need a higher percentage of those 2A fibers to be able to chase down prey, things like that, chase down animals in order to hunt them, versus women. Historically, I don't want to like cross too many boundaries there, but the survival of our race is pretty much contingent on the existence of women. So their ability to sustain themselves, I think, is related to that percentage of type 1 fibers.
Gotcha.
At a more mechanistic level, I think that there might be some impacts when it comes to estrogen. Estrogen being related to like oxidative metabolism and metabolism in general. But I've never seen anything relating estrogen levels to type 1 proportion. But if I think about my thesis where we looked at a middle-aged powerlifter who had been on anabolic steroids, he had 79% type 2s. So one of the highest recorded type 2s ever reported.
Yeah.
That makes me think. So there's definitely a growth capacity aspect of it. Obviously, there's a training capacity portion to that as well. But if a specific type of fiber is going to grow, I think people assume that it's going to be a type 2A. So people have a better association with the growth of type 2As, at least in the acute sense. It seems that at least in the acute sense, type 2A fibers grow faster to make up some of those hypertrophy differences, and maybe type 1s follow after that. But yeah, that was kind of a loose mechanistic interpretation of it. Yeah, I don't have a fantastic answer for that because I've never truly dove deep to try to figure that out.
Yeah, well, perhaps in the future we'll have a little bit more information about that, and that's one of those things you kind of have to, you got to guess, because we don't. There's so much information that we don't have. The data isn't there. So you kind of, based off what I think about this, maybe this is why. So but
we're citing studies, like very few studies with a fiber type in females. But I mean, I think if you talk to any researcher, they wouldn't be scared to admit that we haven't had a lot of female subjects. Comparatively speaking, there aren't many studies out there that are looking to compare men and women, especially when it comes to like performance metrics or just general comparative means, something like what we're doing.
Yeah, my friend Mandy, who is also in the Baylor master's program, she just finished up her PhD at Kansas. Nice. And she was looking at motor unit recruitment differences, which we got to talk about a little bit when I had her on the podcast. But it's one of those things, like you're saying, we just don't have a ton of information on female subjects.
Yeah.
on female research subjects yet. So, but that's changing, which is
Yeah.
great. So
Yeah.
we're starting to get a little bit, here and there, and hopefully over time that'll continue to grow. So we'll have more, like what is her name? I can't think of it right now. Dr. Stacy Sims, I want to say.
Sounds familiar.
Anyway, she's got a book that it's not the book. She's got a famous saying, and that's basically that women are not small men. Like
Yeah.
there's a lot of differences there. So until we have the data on it, we don't know exactly what those differences are, and that information is kind of useful. It's kind of important.
Right.
So
Yeah, I mean, there have been drugs that have been misdosed because the research has been done in men. So
for sure.
yeah, it's definitely an area that at least now people are aware of the deficit that exists. So that's the first step, is recognizing that the deficit exists, and now potentially money can go into avenues of research looking at differences between men and women. Because I mean, the novelty is open, right? You can literally do any other study that's been done before. How does this affect women, right?
For sure. That's so true.
And the methods are already there for you, basically, if you're picking another study. So
yeah, that's the truth. So let's talk a little bit about the practical application. So
yeah, I actually didn't mention the creatine analogs. I didn't talk about.
Oh, sorry. Go ahead. Let's
No,
talk about that.
I didn't get to that. So the creatine analogs, we didn't see any differences, which is really interesting to me. So we didn't see any differences between genders, between groups when it came to total creatine, serum creatine, creatinine. We didn't see any differences when it came to transporter, which I thought was really interesting because some of these things are supposed to be related to the fiber type. So creatine content tends to be higher in type 2 fibers and fast twitch fibers, but yet we didn't see that. Creatine transporter content tends to be higher in type 1 fibers, but even though the powerlifters had a higher proportion of type 1s, we didn't see a difference there. So a lot of these things were really interesting, and I guess that leads me into one of the limitations, or one of the future prospects for it would be, I would want to look at this at a single fiber level where we take a larger sample. And did we potentially dilute some of our findings because we were using a homogeneous, kind of a ground up sample? What if we took individual 2A fibers and looked at the creatine content there, or the phosphocreatine content, the total creatine content? Would that have been different? What if we took the individual myosin heavy chain 1 fibers, or the type 1 fibers, and looked at the order in that regard? So, yep.
Very cool. So for powerlifters and non-powerlifters, the spectrum of athletes, what's the takeaway? What can the strength coach, what can a kinesiologist, what can what's the takeaway here?
Yeah. So our correlations didn't pan out either. So our type 2As and our creatine content had no significant correlation with Wilks coefficient. So specifically for powerlifters, I would say that, number one, we can tell that they're not molecular scale predictors of powerlifting performance. So I don't think that people that are powerlifters need to worry so much about their fiber type. I think that it's probably more important for you to not necessarily pick a program based on what you think your fiber type is going to change to. Also, because there were no differences in creatine content, I think that some people will talk about, this person's a creatine responder, this person's not a creatine responder. And typically that non-responder is someone who tends to have more basal creatine than the average person, or some people that may have a lower level. So I think that in that regard, you shouldn't necessarily be trying to train to increase those types of things either. But it is worth noting that creatine supplementation is probably one of the most empirically backed types of supplementation in sports nutrition. So that may be a means of increasing strength there, but I wouldn't necessarily say that there's a tight correlation with the amount of creatine that someone has and their powerlifting performance. So I think that if we were able to look at something like training capacity, that might be a different story. Same thing with fiber type. There might be individualized responses to how someone reacts to training given their fiber type. There might be individualized responses to a certain type of training, a certain rep range, a certain set number, all these different modalities, cluster sets, things like that, depending on some of these molecular scale predictors, like creatine content, like fast twitch fiber content. But at the end of the day, I don't think that Wilks coefficient and these fiber type and creatine content are going to be very significantly correlated together, or they're not. So
Gotcha. So this in the realm of endurance athletics, the idea of it's everybody wants to know their VO2 max, right? It's the number, right? And it was, when I was in college studying XPhys, like I wanted to get the VO2 max test. I wanted to know what my VO2 was. I was a pretty elite middle distance runner, and sure enough, I had a elite VO2 max score, right? Or number. And it's one of those things that like everybody wants to know it. And then there's some people who base their whole training program around what that number is.
Right.
Like that's the number that they use. And it was actually my college coach who was like not concerned with having anybody on the team do these tests to figure out what the number was. He was like, if you run fast, then it's gonna run fast for endurance, run fast for a long time. If you've got a fast 5K or a fast 8K or a fast 10K, you're gonna have a pretty high VO2. Like it's a cool number, but he didn't find that it was useful to know exactly what that number was. In other words, it didn't dictate the way that he was planning out his training. So do you think that knowing your muscle fiber type and maybe knowing your muscle creatine, content, is it sort of the same thing where it's like, if you're training a certain way, if you're doing certain things, then your muscle fiber type and the creatine content will follow and not kind of the other way around? Like you should dictate your training based off what these numbers are, or you should just, if you're a powerlifter, you get strong and your body will adapt in whatever way it finds to be the most effective based off your training. Does that make sense?
I think that latter thing really resonates really well. So focusing not so much on trying to improve the kind of abstract values, so your VO2 max or your fiber type, which is even more difficult to try to figure out because you have to get a biopsy and all of that stuff. But trying to get stronger, trying to get the more objective outcomes that you can get for your sport. Trying to increase your 1RM is probably going to be, that's the only thing that really matters in powerlifting. Like no one cares about what your type 2 fiber content is or how much creatine you got. You're being judged on how much you can lift on the platform, right?
Right.
It's really cool to be able to profile people, and that's kind of the downfall of fiber type research, is especially because we're looking at athletes and we're cross-sectionally analyzing them. I mean, we can see that these people have a certain percentage of type 2A fibers, and we can say that, well, yeah, like in Dr. Galpin's weightlifter paper, he saw that the more elite, the world-class level weightlifters, they had a higher percentage of those 2A fibers. But that isn't the end-all be-all, right? That doesn't necessarily mean like, oh, I need to have as many 2A fibers as possible. Or that may not mean, or I would assume that it doesn't mean that the best powerlifter in the world is going to have the highest amount of 2A fibers or the highest muscle total creatine content, right? It's probably going to be something. It's going to be a combination of factors. Those might contribute towards it in some very small way. But overall, it's going to be some of those other things that I talked about before. So it's going to be, your limb lengths, your individual leverages, things like that. It's going to be your recovery capacity, which is multifactorial. It's not going to just be related to your fiber type. It's not just going to be related to creatine content. It's not going to just be related to, a lot of people are looking at 23andMe right now, and they're looking at their alpha actinin 3. I did that, and I'm the recessive gene like all the way. So I'm like, oh man, does that mean that I shouldn't be powerlifting anymore? It's like, no, I can still get better, right? I may not be, it's unlikely for me to be a world champion at that, but I think I knew that already. If I'm, if I'm not close to being a world champion already, you're probably not going to be there, right? Especially 28 years old. Yeah.
Yeah.
So I think that's really cool, and I think it's also really cool. I forgot to mention that like the female aspect of, we talked about that too. That you're not pigeonholed into a particular phenotype based on your gender, right? So we showed that females can, with training, they can pretty much have the same content of type 2 fibers as males can, with specific types of training. So I think that's really important to know, that you can kind of equalize the playing field. After talking about how fiber type doesn't really matter, but I mean, that's just talking about how the females don't need to be treated drastically different when it comes to the training stimulus that you're giving them. A lot of the same principles still apply. You just have to tweak them a little bit more based on that. The individual person's, I would say, size is going to be a better predictor of that, right? So maybe you don't give, or a lot of the research is actually showing that you can give females a little bit more volume than males, because they do tend to recover a little bit faster. And I'm not, based on the research that we've done and based on the research that Andy Galpin's group has done, I wouldn't say that that's mediated in large part by fiber type.
Wow. That, I just thought of another thing. Another variable that a lot of endurance runners like to look at is lactate threshold.
Oh, yeah.
Like, I know plenty of people who probably had a higher VO2 max than me, and may even had a higher lactate threshold than me, and I'd still beat them in a race.
Yeah.
Just because at the end of the day, those numbers are great, and they give you a let's say a range for your potential, but sport is all about competition. Sometimes you just got to find a way to gut it out. And I think a lot of times some of the best athletes, when it comes to performance, may not always have the best numbers when it comes to these research measures, or these variables that we can test. Like the fastest sprinter is not usually the strongest person on the track. You know what I mean? So like, there's all these numbers are good and they're really cool, but I think sometimes people get so fixated on stuff that they think is the end all be all, and
you make a good point.
Maybe it's not.
You make a good point there, because it's I think there is some value. Obviously, there's some value in looking at some of these things, looking at lactate threshold, looking at VO2. But when that becomes the end all be all, right? We might need it to be at a certain kind of minimal threshold in order to make sure that we're achieving something. But I mean, even in powerlifting performance, like the best deadlifter isn't always going to be the one who has the best total at the end of the day, right? You can be fantastic at deadlift and you can suck at the other two, and you're not going to win at the end of the day, right? It's a combination of all these factors. It always blows my mind when I see someone that never takes the best. They never take a medal in individual lifts, but when they get the total, they're just all around good at it, right? So kind of mediocrity in all three ends up being fantastic overall. So I mean, that's kind of an abstract way of kind of looking at that. But I think that we want to focus on using the tools that are available to us, and getting the available data and making those informed decisions to be able to create something that's more comprehensive by the end, and not just one thing.
For sure. No, I think that's it, man. For sure.
Yeah.
So how can people do your work? How can they reach out to you if they have questions?
Yeah, I just like talking about research and powerlifting as much as I can. So if people want to reach out to me, Instagram's a good place. So my Instagram handle is stretch_RX, RX like prescription. It's a weird story behind that name, but I've kept it for a long time and my wife won't let me change it. So So that's probably the best way. I know I need to get a Twitter. I keep hearing from people how Twitter is such a great way to like promote your research and all that. But when I have more of it, maybe I'll go out there and do that. Otherwise, I'll just rely on my friends to do that. Um, Other than that, I guess email, but Instagram, I spend way too much time on Instagram. It's probably like my social media vice. So that
Gotcha.
is definitely the way.
Sweet. Well, I'll include that in the show description so that everybody can follow you and reach out to you.
Perfect. As long
With
as they, I didn't lose them already. So
I can't guarantee you're gonna have like, you know, Joe Rogan level, followers after this, but you might pick up a few.
if I'm going to be on Joe Rogan, it's probably not going to be to talk about my research. It's going to be to talk about DMT or something like that.
Do you study that?
Oh, well. So interestingly enough, I actually published a really small review on psychedelics for performance.
No way.
Yeah, no one reads it. I got asked.
Don't tell me too much because we're gonna have to do an episode just about that because
Oh,
I'm very interested in that.
okay. So the only thing I'll tell you is that I actually got a call for an interview with this one reporter from Bryant Gumbel's Real Sports. I was like, why are you calling me? Like, I don't research this stuff at all. It was for a public health class. I just had to talk about something I was interested in, and the topic was the decriminalization of psilocybin mushrooms.
Mm-hmm.
And I just went off on it. Like, whenever I write a review for class, I'm like, if I'm gonna write a review, I'm gonna put effort into it. Even if it's total garbage, I'm just gonna put a bunch of effort into it. And it's like the only paper that exists in the literature that talks about psychedelics for performance.
Wow, that's so crazy because like Rogan talks about it all the time about all these people in Silicon Valley and then all of these MMA fighters who are microdosing.
Yeah,
And
that's why I was exposed to it because my wife and I lived in the Bay Area, so I was a trainer for 3 years, which I didn't mention before, but that one of my clients was really into that stuff, and it was super super intriguing to me. So I did some reading on it, and California, man. So
yeah, we're gonna have to do an episode about that because that sounds incredibly interesting to me.
Well, I don't do research in it, but I just like talking about it. So we can.
Well, we're gonna talk about it.
Yeah.
Yes. Awesome. Sweet. Well, dude, thank you so much for taking the time to do this. This has been excellent.
Yeah, of course.
I've really enjoyed it. I've learned quite a bit, especially a whole bunch of stuff about Type IIx that I thought I knew.
Yeah.
That now I'm like, oh, who cares about Type IIx? I don't care.
What I learned this is like the first ever podcast that I've done with someone, so I thank you for this opportunity because it's been great to watch myself like stumble my own words and like multiple times where I'm like, you asked me for the results of the study and then I forgot to talk about this and then I forgot to talk about that and I'm like, well, I guess I gotta re-mention that real quick.
Yeah, man. That's the nice thing about this. Like, there's no time limit. Like, we can go back. We can add to
out
it.
all the parts where I make myself sound like an idiot. That sounds good to me.
I try to do as little editing as possible. So I honestly don't think I'm gonna have to do too much here. Like
that sounds good.
Yeah, man. You said ATPase, so
yeah, that was my one win of the day.
I'm not editing out anything. This has been awesome, man. Alrighty, y'all. Thank you so much for watching and listening. Be sure to go follow Steven, and if you have any questions for him, definitely reach out to him. And stay tuned for next week's episode. And also, stay tuned for when we get him back on to talk about some more fun stuff. Alrighty. Adios.
Stuff.